Welcome back friends, welcome back in another video tutorial for microbiology with Shagnik. So in this video tutorial we will going to discuss our second part of ubiquitin mediated degradation of protein. So in the first video of ubiquitin mediated degradation of protein, we have discussed the detailed concept of the structure of proteasome, right? So, and in today's video we will going to talk about how ubiquitin is attached with protein. Okay, so before going to enter in this today's video, I want to discuss a quick recap of the previous class. So in the previous class we have talked about the structure of proteasome, right? We have talked about the two parts of proteasome. One is 20s core particle, another one is 19s regulatory particle. The function of 19s regulatory particle is to recognize the proteins attached with ubiquitin, right? And also to cleave that ubiquitin so that this particular ubiquitin can be reused or can be recycled, right? And the 20s core particle was the main part where ATP dependent degradation of protein will occur, right? So today we will going to learn how this ubiquitin will going to attached with a particular protein, okay, by which the proteasome can recognize that yes, I have to degrade that particular protein. Although attachment of ubiquitin is not means that this protein will going to surely be degraded, okay, because there are various type of ubiquitination that occurs. For example, monoubiquitination, multi-ubiquitination, polyubiquitination, and there are different meanings of them. So now we will going to talk about how this ubiquitination or ubiquitylation occurs. Okay, so first, that this ubiquitin is a eukaryotic protein which is a highly conserved eukaryotic protein and consists of 76 amino acid. Okay, now this ubiquitin will going to attached with the lysine residue of the target protein by its C-terminal glycine. Okay, and there are various type of ubiquitination that can occur to a particular protein. As I have told you, the particular protein can be monoubiquitinated, the particular protein can be multi-ubiquitinated, the particular protein can be polyubiquitinated. Okay, and in case of degradation purpose, a particular type of protein is mostly polyubiquitinated. Okay, so now we will going to talk about how this ubiquitination occurs. So as you can see in this picture, there are various type of ubiquitination that can occur. For example, monoubiquitination, multi-ubiquitination, polyubiquitination that can occur. Okay. So now let's move on the next slide. So the process of ubiquitination. So the first step is the activation of ubiquitin. Okay. First of all, the ubiquitin should be activated, and this activation of ubiquitin is done by E1, that is ubiquitin activating enzyme. First of all, as I have told you that ubiquitin is a eukaryotic protein of 76 amino acid residue, so obviously there should be some free C terminals, right? So C terminal means free COOH group in ubiquitin, right? So this E1 will going to interact with that free C-terminal residue of ubiquitin. Okay, and this interaction is a covalent interaction. Okay, E1 with its SH group, that may be due to presence of cysteine residue in that particular enzyme, will going to interact, which is obviously a covalent interaction with its free C-terminal residue of ubiquitin. Okay, so this is how activation of ubiquitin occurs. Okay, so again, ubiquitin have free C terminal in, that is the free COOH group, and E1 will going to interact with this free COOH group with its SH group. Okay, and that's how activation of ubiquitin occurs. Next step is transfer of ubiquitin from E1 to E2. Okay, and this is facilitated by cysteine residue of E2. Okay, that means from E1, that activated ubiquitin will be transferred to the cysteine residue of E2. Okay, and E2 is the ubiquitin conjugating enzyme. Now in the third part, that is the ligation of ubiquitin to target protein. Here what happens? This ligation of ubiquitin is done by E3, which is ubiquitin ligase. What this ubiquitin ligase will do, it will take the activated ubiquitin from the cysteine residue of E2, and this activated ubiquitin will be transferred to the lysine residue of the target protein. Okay, and there will be a formation of isopeptide bond. This is very very important for any kind of MCQ exam purposes, right? That is, ubiquitin is attached via a isopeptide bond formation with the lysine residue of the target protein. Okay, you have to remember this. And once one ubiquitin is attached with the target protein, now this entire thing will going to be very very useful for polyubiquitination. As I have just told you that ubiquitination may be of various type. It may be monoubiquitination, multiubiquitination, polyubiquitination. So for forming a polymer of ubiquitin, one ubiquitin attached with the target protein is very very important. Okay, because as I have told you, the ubiquitin which was already attached with the target protein via first ubiquitination should have some free should have some free internal lysine residue. Okay, as because ubiquitin is also a protein, right? So there should be some lysine residue, and it was observed that ubiquitin has 7 lysine residue. Now this free internal C terminal, like not C terminal, this free internal lysine residue will going to interact with the another C terminal of ubiquitin. Okay, so that's how the entire thing is done. Once again, once one ubiquitin is already attached with the C terminal of ubiquitin, this ubiquitin should have some lysine residue, right, which is for now free. Now this free lysine residue will going to interact with another ubiquitin. That's how the entire polymerization of ubiquitin occurs. Okay, this is one kind of polymerization of ubiquitin that can occur. Another one is that particular ubiquitin can form a covalent interaction with the N-terminal alpha amino group via a peptide linkage to yield linear polyubiquitin. That may also occur. Okay, so that is the two type of ligation that occurs. So if we try to sum up the entire thing, so what happens if you watch this picture? So with the help of ATP, E1 will going to take the ubiquitin and activating it, right? Then this transfer of ubiquitin occurs from E1 to E2, right? Or more specifically saying, the cysteine residue of E2, right? Then E3 will going to transfer that activated ubiquitin from that E2 to the substrate, right? Substrate means here the target protein, right? In the target protein, there is a lysine residue where the ubiquitin mainly binds, right? That is the entire idea of how ubiquitination is achieved. Okay, so now there is a small region in the target protein which is recognized by E3 ubiquitin ligase, and this thing is important for substrate binding and also ubiquitination. This is degrons. Okay, this degrons things is necessary so that the E3 ubiquitin ligase can recognize the target protein. Now, a polyubiquitinated protein is degraded into short peptide by an ATP dependent process mediated by proteasome, and we have also seen this when we have talked about the MHC class 1 molecules endogenous pathway, right? We have seen this thing earlier. So that is the idea of how the ubiquitination occurs. Once again, first of all, the E1, that is ubiquitin activating enzyme, will going to interact with the free C terminal end of the ubiquitin, right? That is how activation occurs. Then transfer of activated ubiquitin to the cysteine residue of E2 occurs. That is the transfer of ubiquitin. then ligation occurs, where E3 will going to take that ubiquitin from the cysteine residue of E2 and it will deliver it to the lysine residue of the target protein, right? Now, once one ubiquitination occurs in the target protein, it will facilitate polyubiquitination, right? Because that particular ubiquitin should have some internal lysine residue which is for now free. It will going to interact with the C terminal of the next ubiquitin residue. And interestingly, ubiquitin has 7 lysine residues, so this particular theory is appropriately applicable, right? So another type of theory that is also present that depicts that in this way also ubiquitination can occur, that is ubiquitin may also attach with the N terminal alpha amino groups. That's how the linear ubiquitin or linear polyubiquitin chains are formed. Okay, and most importantly, each ubiquitin ligase recognizes the target protein by seeing the de Grand's regions. Okay, so that is how the ubiquitination occurs. So I hope the concept of how ubiquitination occurs is very clear to you. Now let's talk about a little bit about the E3 ubiquitin ligase. So E3 ubiquitin ligase are of two types. One is ring family E3 ubiquitin ligase, another one is HECT ubiquitin ligase, and these two family are generally characterized depending upon their conserved structural domain and how they transfer ubiquitin from E2 to the target protein. So first, this ring protein will going to transfer the target protein from E2 to the target protein directly. Okay, again, this ring protein, what this ring E3 ubiquitin ligase will do, this ring E3 ubiquitin ligase will going to transfer the target protein from the E2. So I am sorry, so very sorry. So what will occur, that this ring protein will going to transfer the ubiquitin from the E2 to directly the target protein. That's what I am actually want to teach you. So that is the idea, that the ring family will going to catalyze the direct transfer of ubiquitin from E2 to the lysine residue of the target protein. But it is quite different in case of the HECT family of E3 ubiquitin ligase, because in case of HECT family of E3 ubiquitin ligase, the ubiquitin will be first transferred from E2 to the active site of the E3, and then from the E3 it will be transferred to the target protein. That is the idea how HECT works. Okay, and in that particular case, in the E3 there should be cysteine residue in the active site. So among the ring family E3 ubiquitin ligase and HEC3 ubiquitin ligase, ring family is the most common, and we have already seen one type of ring family E3 ubiquitin ligase in our cell cycle videos, that is SCF complex, right? SCF complex consists of three things: SKP1, CAL1 and a box protein, right? So CAL1, also known as Qulin1, is a scaffold protein, and F-box protein is another type of protein, and in between them SKP1, that is S-phase kinase associated protein, will act as a bridge. That is the entire structure of SCF complex, and this complex may also contain some ring protein also, which are small. They are known as RBX1. So that is the idea of SCF complex. So again, SCF complex consists of three things: SKP1, CAL1 and F-box protein. So CAL1, also known as Qulin1, which is a scaffold protein. In between CAL1 and F-box protein, there should be SKP1 as a bridging material. Okay, and this SKP1, the full form of SKP1 is the S-phase kinase associated protein. Along with that, there should be some small ring proteins also, and they are known as RBX1. So that is the entire concept of SCF complex. So this is the end of our ubiquitination of protein, how ubiquitin is attached with the protein. We have also talked a little bit about how the polyubiquitination occurs, and we have also talked about the structure of proteasome, right? So this was the end of this video. So I hope these two video will be helpful for you to understand the entire concept of ubiquitination. So thank you for listening to this class.